Hey, it's Mr. Shrum, and I've been getting a lot of questions about these course activities, PBL activities, unit activities, all that jazz. And I'm just going to walk through Unit 2, photosynthesis and cellular respiration, and go through the activities and explain what I'm looking for, explain why it's asking for the things it's asking, and how much I am willing to accept other things on these activities. So here we go. The first one in Unit 2, course activity, aerobic and anaerobic respiration. Let's check it out. In this activity, you will use the results of an experiment to explain the relationship between aerobic and anaerobic respiration. It's a science class, so obviously we're going to talk about experiments and what the results of experiments can tell us about the natural world. Introduction. When you bite into a piece of bread, a soft piece of bread, you're enjoying the work of tiny unicellular fungi called yeast. Bread is made by mixing flour and water and then adding yeast to increase the bread's size and softness. Yeast makes the bread rise or increase in size through respiration. And this is a process where organisms obtain energy from glucose and other carbon compounds. Many organisms need oxygen for respiration, but others can produce energy in environments without oxygen. Aerobic means with oxygen, anaerobic means without oxygen. And anaerobic respiration, where there is no oxygen, is seen in the process of fermentation. The major types of fermentation are lactic acid and alcoholic. In lactic acid fermentation, molecules convert to lactic acid. Lactic acid fermentation often occurs during strenuous exercise. So this is actually occurring inside your body. Your body's creating lactic acid, and that is why you feel this burning sensation if you're working really hard, sprinting, or any very strenuous, very physical activity. You will build up some type of lactic acid in your body. Yeast uses alcoholic fermentation, converting the glucose in the bread dough to carbon dioxide and ethanol. Escaping carbon dioxide bubbles cause the dough to rise, while the ethanol evaporates during the baking process. So when you're baking bread, it's not as if it's alcoholic bread. That's not what's occurring. It's part of the process. And by the time the bread is ready, the CO2, the carbon dioxide bubbles, cause the bubble to rise. And then all that ethanol has disappeared. It's evaporated into another form and is no longer in the bread. Anaerobic respiration is important to humans for several reasons. If you exercise for a long period of time, your muscle cells become deprived of oxygen. The muscle cells then perform lactic acid fermentation to produce the required energy. So your muscle cells, they don't have oxygen anymore. And then this lactic acid fermentation is required to come up with the energy of whatever exercise or activity you are doing. In the food industry, yeast and other microbes that respire means breathe, essentially, anaerobically, are used to make food and beverages. In this activity, you will test the ability of yeast to respire under different conditions. And you see here we have two lovely people exercising. strenuously. All right, so this is an augmented reality. And if you have a smartphone or tablet computer, you have the option to test the respiration of yeast using augmented reality. And that stands for AR. AR stands for augmented reality. And in this exploration, you'll complete a virtual lab using the built-in camera on your mobile device. This AR exploration is optional. Ask your teacher whether you should complete the exploration and related worksheet. I say try it out if you have a smartphone and you're wanting to do this exploratory activity. And I'll show you what to do. But this is an optional assignment, and you can download the app. If you have Android, it's on Google Play. If you have an Apple device, some type of iPhone, the App Store looks like this. And the app is called Edmentum AR Biology. It looks like this. And as you work, you have these AR worksheets to record your work if required by your teacher. I'm not requiring this, but I think it is fun if you are willing to download an app and just test things out. That's what science should be about, is experimenting, having fun, doing new things, and seeing what you can do. Give yourself plenty of space to move around. So it's just like, I don't know, playing Wii or PlayStation Move. Like you're gonna need some room to move around. And don't do this in any weird hazardous areas where you can break things. Avoid covering your device's camera. Actions that cause you to lose progress, like letting your device go to sleep, clicking the back arrow during AR, closing and opening the app in the middle of an exploration. Menus on the side of the screen contain helpful information. Tap the arrows to expand the menu. So here's the accompanying worksheet, aerobic and anaerobic respiration. If you want to use the AR activity. And here, yeast is a live organism. When yeast undergoes respiration, it produces carbon dioxide gas, CO2. During the experiment, the gas appears as foam at the top of the liquid. So keep that in mind. And then scoring, this will be the regular graded activity task in the course activities. And part A is our hypothesis. In this activity, you will test the ability of yeast to respire under different conditions. Jared, a student, is working on an experiment. He plans to measure the amount of CO2 that yeast produces when it is fermented in 5 different glasses. And the glasses have the following contents: yeast in lukewarm water, yeast, sugar, and lukewarm water. Lukewarm means it's not exactly hot or cold water. It's sort of a room temp, maybe slightly above room temperature. Yeast, sugar, and hot water, yeast, sugar, and cold water, yeast, sugar, and lukewarm water covered with plastic wrap. All right. The formation of foam is evident that respiration is taking place because CO2, carbon dioxide, is a product of respiration. One end product of respiration is CO2. Because CO2 produces the foam in the glass, Jared measures the foam to see if respiration has taken place. Do you think the amount of foam in the glasses will differ? Write down your prediction. So basically, which do you think will react and cause some foam to manifest? Glass A won't foam at all because there's no sugar. And this process requires sugar to take place. And without this sugar, there will be no glucose molecules for the yeast to act upon. Yeast needs glucose. The yeast won't produce any foam in the covered glass since most organisms need oxygen for respiration. The yeast will produce the most foam in hot water. That is the example prediction here. So you need sugar, you need glucose, you need oxygen for respiration and fermentation to take place. Part B is about the design. So in order to perform the experiment, Jared will follow these instructions. And they're listed out very meticulously in detail. Put 1 or 2 ice cubes in 1 cup of water. Chill that cup of water in the fridge for 1 to 2 hours. Remove any remaining ice when taking the water out of the fridge. This cup holds the ice cold water. Heat up 3 cups of water in the microwave for 15 to 20 seconds until they reach 100 degrees Fahrenheit on the thermometer. If the water is too hot, wait until the temperature drops down to 100 degrees Fahrenheit. These cups hold the lukewarm water. Heat the last cup of water in the microwave until it reaches 212 degrees Fahrenheit. This is the boiling hot water because 212 degrees Fahrenheit is the boiling point of water. Line up 5 glasses and label them A, B, C, D, and E. Then put the following contents in each glass. So we got the lukewarm water. We got the lukewarm water and tablespoon of sugar. Boiling water, tablespoon of sugar. Cold water, 1 tablespoon of sugar. Lukewarm water and tablespoon of sugar covered with plastic wrap. Stir the contents, make sure the sugar dissolves in the ones that have sugar. Then you're going to add a packet of yeast to each glass. Wash the spoon and stir in the yeast. Cover the top of glass E tightly with plastic wrap. Wait 15 minutes, then measure the foam level in each glass in centimeters as shown in the image. We have foam, water, and yeast here, and then you're measuring the foam from the rim of the glass and the top of the water. We're going to take measurements of the foam level every 15 minutes for 60 minutes. So 15 minutes, take 1, 15 more minutes. That's your second one. 15 more minutes, 3, and then 15 more minutes, 4. What is Jared testing in this experiment? Well, we are testing the amount of respiration that actually occurs. And we're measuring that by measuring the amount of foam produced in each cup. More foam means more respiration. And foam is the result of the production of CO2. That's where all the bubbles are coming from. Covering glass E with plastic wrap prevents oxygen from entering the glass, so Jared can test whether anaerobic respiration occurs. Anaerobic respiration does not require the presence of oxygen. And he can test whether anaerobic occurs by removing the oxygen from the cup. And that is what the plastic wrap is doing. Now we're pairing up the glasses and the variables. So what do A and B have in common? There's sugar, or you're testing the variable and comparing between the two. So between A and B, you're seeing if the sugar has something to do with it. Between B, C, and D, you're changing the water temperature. B and E, we're looking for the type of respiration: oxygen, aerobic, non-oxygen, anaerobic. And here's an easier explanation right there. A and B, presence of sugar. B, C, and D, water temperature. B and E, type of respiration. And in part C, we are given the table that Jared recorded every 15 minutes for an hour. So you see A starts out with no foam. 60 minutes later, still no foam. B, we got some foamage. C, no foam. D, a little bit of foam, but not very much. And then E, we have foam. So for the example prediction, it says, I predicted that the yeast would produce the most CO2 in hot water, but that was wrong. It didn't produce any foam. However, my prediction that the yeast wouldn't produce CO2 in water alone was correct. So sometimes our predictions are not correct, and that's okay. That's part of science. Hypothesis, we're looking at what we know and making predictions off of what we know and trying to explain natural phenomenon. And sometimes you're not right, but the more you learn, the better your hypotheses will be. At lukewarm temperatures, the yeast was able to produce much more CO2 from the sugar compared to cold temperatures. At higher temperatures, the yeast didn't produce any CO2, and that suggests the higher temperatures are actually killing the yeast. I concluded that the yeast needs warm temperatures to thrive. So warm, not boiling, and not too cold. Yeast can perform both anaerobic and aerobic respiration. Does this experiment demonstrate that? Yes. It produces, or the yeast produced equal levels of foam, whether it was uncovered, so it had access to oxygen, or it was covered and it did not have any access to oxygen. So it was able to do both. Yeast is added to bread dough before baking. Use your experiment to explain how respiration of yeast is important for the baking process. Well, within that bread dough, we have carbohydrates. We have sugar molecules and glucose molecules. Those carbs can be broken down into those glucose chains and molecules. This process gives the yeast a food source and allows them to produce CO2. The bread dough then rises, and without yeast, the bread would be flat. Some of our human cells can perform anaerobic respiration for limited amounts of time, such as during periods of heavy exercise. Use the results of your experiment to explain how anaerobic respiration could benefit human survival. Well, in this experiment, it shows that energy production or respiration occurred in yeast even when little oxygen was present. During exercise, the lungs must be working hard to take in oxygen. And then that oxygen is circulated to the body parts that are needing more oxygen. The ability to perform anaerobic respiration for short periods of time allows humans to sustain activity, even when oxygen is limited in the body. And that is the end of that course activity. I hope that was helpful. And let me know if you try the augmented reality. I'm going to try with my phone and screen capture it after I update things. I've kind of slacked on updating my phone. But hey, sometimes I don't love the updates on everything. But anyway, that's it for this first course activity, and I'll talk about the next one later. But until then, have a good one, and I will talk to you guys later. Bye-bye.